Good. Well, it is an honor to introduce Jim McGaugh, who is a man who really needs no introduction here. He is the founder of the Center for the Neurobiology of Learning and Memory and the founding chair of the Department of Neurobiology and Behavior at the University of California at Irvine, which was the first of its kind and only of its kind at the time. He was the founding father of the study. Let's see if I can fix this. All right, better? Good. Okay, so he was the founding father of the study of the emotional modulation of memory consolidation, and his seminal studies in the 1960s, where he found that he could give a drug after training and not just impair memory, but actually enhance memory, really turned the field on its head. And he had a hard time publishing some of that work early on. But since then, looking back, if you think about the impact that work had on the field, it's really unfathomable. I mean, it's just the extent is incredible to think about what we've learned about cellular and molecular mechanisms of memory because of the window that he opened. So Scoville and Milner showed us that they showed us where we could look in the brain for memory, but he showed us when we could look in the brain for memory. He is also a former dean and vice chancellor of UC Irvine, and a member of the National Academy of Sciences, and he has over 460 publications. Not that I think he counts, but that really doesn't define him. For those of us who are fortunate enough to call him our mentor, he's so much more than that. He's been a mentor to 200 undergraduate students in his lab, 25 Ph.D. students in his lab, 75 postdocs and visiting scholars. And I suspect that he's been a mentor to many of you, even if you weren't in his lab. I'm sure that he has said something to you or you've learned something from him that has profoundly affected the way you think. So the way he thinks is very exceptional, I think. He is very clear-minded, and his experiments and his ideas are elegant and conscientious, and he tries to share that with anybody who he can. And so he's just a very generous teacher in this way. So I've invited him to come to visit me and my university, the University of Texas at Dallas, many times, not to ask him to give a big public lecture for a hefty price, but actually to be in front of as many students as he possibly could be in front of, because the way he thinks is something that you can't really learn from a textbook or journal article. It's just something very special that he's generous and sharing. So I'm sure that, let's see, I don't want to forget anything. There are too many things to say about him, so I only have 2 minutes. I'm in a hurry. Let's see. All right, so, okay, so my final point. I think that the thing that makes him most exceptional and most extraordinary is his emotion. So he is a man who studies emotional modulation, and I think it's very interesting that he uses so much emotion in his research. He just completely infuses it with joy and with gratitude and with awe and amazement. And so when you're in his presence, you can't help but to be inspired to do the best job that you can do and to be thankful to have the job. So now I will turn it over to Jim, who after 60 years, I think it's been almost 60 years since he entered Berkeley as a PhD student, he's now blazing new trails, studying this highly superior autobiographical memory. So I will turn it over to him now to share that with us. So please give him a warm welcome. Oh my goodness. I wish I could say
more. After that, I can only fail. I mean, after all. Well, welcome to all of you. I am delighted and surprised to see all of you here. When I was told I was going to speak at 8 o'clock in the morning, and knowing the usual habits of my colleagues and graduate students and so on, I thought there'd be several of us here and we could just have a little seminar, half a dozen. But now I see the room is crowded. Thank you all for coming. And thanks to Mike and Manuela and the crew that they put together for organizing this magnificent meeting. I think we should thank them right now, please. And I was honored to be asked to give the first talk, and as I say, I accepted because I thought nobody would be here, but now that you're here, I'm obligated to say something. A little over 35 years, Norman Weinberger and Gary Lynch and I got together and said, it's time to give an increased focus to the study of learning and memory, not just as a side thing, but really a deep focus on it. And so we established, with the approval of the Regents of the University of California, the first center for the neurobiology of learning and memory in the world, and it still exists today. We thought it was important, and we think that it is terribly important. Why? If you think about it, most of us just go about our daily work working on some kind of a problem, either in learning or memory or related to it, because that's what we do. But I want you to stop and think back for a moment about what it's like. Our lives are about a second or less long. That's it. And by the time you think about it, it's now past. And now you think about it, you can think about the next second, and by the time you do it, it's over. Isn't that incredible? We live in a world about 1 second or less long. And the glue that makes it possible for us to exist consists of the processes underlying the formation and the maintenance of memory that create substance to our lives. That's how important it is. So I want you to think about it and congratulate yourselves that you're working on learning and memory, because I regard it as the most important problem in science. Because without it, there would be no science. Without it, there would be no we. It is just critical to our existence. Memory is amazing. And of course, lots of folks over the course of the centuries have known that it is amazing. Back in the olden days, at the time of the early philosophers, and I was not there at the time, the philosophers wondered about how this all happened, and I'm just going to give you just a quick notion here. The Egyptians thought about it, and they thought that these processes somehow were located in the heart. Now, that should be obvious to you, because when you learn something very well, at least when you were younger, you said you learned it. You didn't say, I learned it by spleen. You said, I learned it by heart. Thanks to the Egyptians for that. You learned it by heart because the Egyptians said it was probably there. And as an aside, by the way, one of the reasons I'm told, or I learned, that the early people thought that the heart was so important, because that when they were in battle and people were badly injured, they could still see that the heart was beating. And so it had this feeling of life character, and they said that must be where all of the processes are located. Plato got it right, but nobody paid attention to it, because by the time Aristotle got there, it's back in the heart again. And Leonardo da Vinci, whom we honor and respect for so many reasons, was more modern than that. By that time, they had discovered the ventricles, and so it was in the ventricles. And I thought a lot about that, and maybe it's because at that time, they knew a lot about the movement of fluids. I mean, they were building canals and doing all kinds of things, and so the mechanics of fluids were very well known at that time, and that may be why Leonardo da Vinci, who was an inventor, as you all know, not just an artist, but an inventor, that's probably why he located it there. Now, I want to devote most of my time to somebody who is usually ridiculed, and that's Franz Joseph Gall. How many of you, honestly, have ridiculed him, made fun of phrenology? Ha, ha, ha. Come on, raise your hands. Raise your hands. All right. I'm going to change your minds. That's the basis thesis that I have here. Friends, Joseph Gall, in the early 19th century, said once and for all that memory processes are in the brain. And that was settled not until the early part of the 19th century. It was as recently as that finally it was decided, okay, it's not the spleen, it's not the toe, it's not anything else, it's up there. And he when he was a student in school, he had trouble memorizing. And he looked around and he saw that the kids in his classes who were really smart and could memorize very well had a forehead that protuberated. It stuck out a little bit like that. And he got the idea that the reason that they were so smart is because they had a bigger region of this head, and that got him thinking about it. And he thought an awful lot about it. He was an anatomist of the highest regard. He was no fool. He thought about it. And here's the more modern version, a guy named Fowler popularized it. And by the way, it got into circuses and carnivals and so on. That's why we laugh at it, because you feel the bumps on people's head and tell them who they are and so on. But that's not what it was about. I'm going to give you the basic assumptions. First, the brain is the sole organ of the mind, and that was settled in the early part of the 19th century and not before. Secondly, and I want you to remember this, the brain contains many independent regions. He was the first person to say that. The differences in intellect are due to differential development of brain regions. How many of you here would agree with all three of these? Come on, raise your hands. If you don't raise, you're asleep if you don't raise your hands. Okay. Now here's where we might have a little problem. The shape of the skull corresponds to the shape of the brain. Is that a ridiculous hypothesis? That's not a ridiculous hypothesis. I mean, the brain is in there and the skull and so on. And up there, everything is okay. And as a matter of fact, you might propose that today. Everything I've told you now would be satisfactory as a proposal today to think about brain and the location of cognitive processes. Now, the problem was that he proposed that the size of the skull regions assess the propensities of the underlying brain. And we might question that. You might say, well, look at the skull, is that going to tell us what is there? And that's where we came apart. And that's why he became to be seen as a fool, because people said, oh, you just put the fingers on the head and, oh, he's telling children here. He had a widow that he knew quite well, and for a reason he didn't tell us, he said the back of the head here is the locus of amorous tendencies. Do not look at the back of heads right now, please. So it became kind of silly, but the fundamental hypotheses, except for this last one, which is questionable, are the ones we have today. But he became known as a fool, and we don't talk about him. He's not prominent in our thinking. He had that silly stuff about the skull, and so now let's get on with real science. I'm telling you, I view him as a real scientist who had ideas, and the technology of the day limited his development of his thinking. He didn't have the tools that we have today, nor did he know how to get those tools, because he was just at the beginning of the scientific study that we know. Now here is the key thing that he and his student wrote early part of the 19th century, and I want you to note this. It is certainly impossible to deny the mutual influence and dependence of the different brain organisms. You can't deny that. However, there's a great difference between the correct assertion that the different organs exert a mutual influence upon each other and the incorrect one that each part does not exert its particular function. You got different regions of the brain, each one has a function, and you cannot deny that they interact. That's what a lot of us in the room do in our studies. We look at regions of the brain and their interactions with other regions of the brain, and the idea underlying that came from Gall in the early part of the 19th century. But he's a fool. You see, and we laugh at him because you feel the bumps on the head. Well, partly he's a fool because he was followed up by Florenz, who actually was on a mission to destroy Gall. He hated him, and so there was a battle between the two. And Florenz had pigeons and chickens and things of that kind, and he was one of the first persons to make brain lesions in the surface of the brain, and did a lot of studies, and he concluded that all the intellectual functions reside exclusively in the cerebrum, in agreement, but they reside there coextensively and without any one being separable from the other. It's one big glob, and Florenz won the battle. That view held almost until 1950, from the time of 1842, almost 100 years, and I'll explain why. There was somebody I'm leaving out here, Franz. How many of you been on the UCLA campus and seen Franz Hall? Any of you? All right, Franz was the teacher of Carl Lashley, and he did experiments early on, then followed up by Carl Lashley, with which most of you are really deeply familiar. Carl Lashley wanted to discover the locus of memory, and he trained laboratory rats on mazes, and he made lesions, either small ones or big ones, before the animals were trained, or in other groups of animals, he trained them, and he made either small lesions or big lesions, and studied their memory of the maze training. And what he found was that he couldn't locate memory. These are all cortical lesions. He couldn't find it any place. And so essentially, he confirmed what Florenz had said, and he did not confirm anything about what Gall had proposed. And so we know about Lashley, and we're puzzled by it. So he studied the effects of brain lesion on memory, and he ended up with the notion that there's mass action and equipotentiality, which is really a confirmation of Florenz. And that gets us up to 1950, which is amazing. And I was a graduate student just 3 years later, and so I grew up on Lashley. That is, when I took learning and memory, I learned about Lashley. I did not learn about Franz Joseph Gall. And then, so we're up to 1950, and then there was a review written in the Handbook of Experimental Psychology, a long review summarizing all of this stuff by Clifford Morgan, who was the major physiological psychologist at the time. And he said in 1951, why there is a correlation with mass, but not the locus of the lesion, is a vexing problem. Here, let us simply note that there is no simple localization of memories in different parts of the brain. That's in 1951, a long period of time since the early 1800s, and Florenz won the battle at that time. Now I'm going to take you back to the 19th century. We got all the way up to 1950. I'm going to take you back because I've left out some other important influences. Ribot. How many of you have heard of Ribot? Good. Wrote a book on diseases of the memory, published in 1882, and he gave us the early indication that the loss of memory varies inversely with time between an experience and brain injury. That is, if you have a brain injury, you're going to lose the most recent memories, not the older one. And that has been verified in many subsequent studies. But this is a very important discovery. It didn't quite fit in with things, so it's sort of sitting on the shelf there, waiting for later discoveries. Hermann Ebbinghaus in 1885 produced the first experimental studies of memory. And if you've studied verbal learning, this may bore you, but the point was that many philosophers at the time and many other people at the time said that human cognition was too complicated and too important, could not be studied scientifically. It's too ephemeral. I mean, the brain is complex and memories are ephemeral. You can't study it scientifically. And Ebbinghaus says, the hell you can't. And he set out to do the very first experimental studies of memory, and he put memory on a scientific basis. And that was a very, very important contribution. There was no physiology there, but he settled once and for all the issue as to whether memory could be studied scientifically, and he showed that it can, and just beautiful experiments. Those of you who have not read the English version of his book, I direct you to it, because you see he applied statistical methods to it. I mean, it's really quite a gem of science. That's Ebbinghaus. William James, in 1890, wrote this book, which is one of the most beautifully written books in the history of the world, Principles of Psychology. There's no experimental science in it, but there are all kinds of important observations made by William James. For one thing, to anticipate what's going to be coming, his chapter on habits was in a different place than his chapter on memory. He separated habits from memory, which is very perceptive on his part. He also distinguished between primary memory and secondary memory, that is, recent memory and lasting memory, or what he called memory proper. So here you have an anticipation of ideas that came later. We don't only have memory, but we have different kinds of memory. According to William James, we have habits, we have recent memory, and we have long-term memory, a beginning of the separation of these processes in 1890. Unfortunately, it did not influence any experimental work at the time. It's just sitting there in history to show that he anticipated much work that came later. One of my favorites, those of you who know my work would know, was Mueller and Pilsaker, who in 1900 proposed the memory consolidation hypothesis, and you know what that is, that memories, when they're first established, take a period of time in order to become completely fixed, and they did the first experiments to demonstrate that. That was in 1900. So these are just ideas that were developing along the way in this period of time. Now, the next half century, aside from Lashley, had nothing to do with any of this. It took a different direction because we start with the experimental studies of learning and memory in laboratory animals, not with a neurobiological focus, but just what is the nature of the learning. And the issue that is focused here, when we and the animals learn, is everything based on the acquisition of responses, or is there cognition? Now, today, that's kind of a silly question because we know the answer, but this was an argument that was fought vigorously and vociferously for 50 years. So we have Pavlov in 1900, who discovered the conditioned reflex. As you all know, he was studying used salivation because he was studying the mechanism of digestion. And during the course of this, he discovered that animals would begin to salivate when they saw the signs that were delivered before meat powder was put on their tongue. And he saw that there was an association then between the stimuli out there and the response, and he gave us classical, or what we call Pavlovian conditioning. Pavlov then wrote a lot, and he said, this is the fundamental learning. This is what learning is. All learning, all education, everything is this kind of conditioning, and it's all in the responses. That was a hard view, and that was his view. It's not just a technique. It's not just Pavlovian conditioning as here's what you do to train animals. His view was that is the nature of our learning. It's the way that the dog learns to salivate when sees a signal before stuff is put on the tongue. So that was Pavlov. We have E.L. Thorndike, who is, how many of you have heard of Thorndike? Maybe, it's wonderful, wonderful. Who gave us instrumental conditioning, or reward conditioning. Usually people attribute that to Skinner, and I do not. It's to Thorndike, who, as you know, put cats in a box, and they had to learn to press a lever or pull on a chain in order to get out and get a little drink of milk. And what he found was, to his satisfaction, that the reward was essential for the learning. Rewards strengthen stimulus-response connections. So animals see something, they make a response, they get a reward, and the reward acts backward to strengthen the connection between those. And that was his view. Once again, it's stimulus-response learning, and all learning is the learning of responses. So it's like Pavlov in that sense, but a slightly different twist on it because reward became essential. So that was those are strong views that started in around the turn of the century. And slightly later then, with a lot of influences, which I have to skip over, including Gestalt psychology, which was very influential at the time, we have Edward Chase Tolman at Berkeley, whose major work was 1920 to 1950, and Truth in Advertising. I was a student of his, so there's a slight bias here, and you can discount that if you want. Tolman had a very different view. His was a cognitive view, that animals can learn what leads to what. That is not, the responses are an outcome of what is learned. It's just that learning doesn't consist of responses, it consists of acquisition of information, which can then be displayed in behavior. And that was laughed at, because after all, you have an animal sitting in a maze, it's at a choice point. From one point of view, the animal's about to execute a well-learned response, and from the other point of view, and cartoons were made about this, you have an animal at the choice point buried deeply in thought. You know, what am I going to do here? Well, Tolman thought they were buried deeply in thought. He thought that there were cognitive processes in action here. The earlier view, it's only responses, and then Tolman said, well, wait a minute, let us attribute some cognition to the animals, and he did a lot of experiments, basically learning what leads to what. I'll just show you one, classical one. You see at the top, the animals are in a tea maze, and they learn, and they turn to the right, and they get food, right? Now you can ask the question, did they learn, and it's in a room, did they learn a turning response, or did they learn that food is over in this part of the room? So what he did was turn the tea maze around, started from the other end, and ask which would you like to go? If they learned a response, they should turn to right, as they always did, but if they've learned where the food is, they should turn left. So this is a very, very simple task, and it turns out that in most experiments, the animals went to the place, they did not learn the response. But is that all they learned? Is it possible that they learned both? Is it possible that we can learn responses and that we can learn cognitive things as well? Tolman finally said yes. He wrote in 1949, there is more than one kind of learning. And without giving the details, Mark Packard in my laboratory a number of years later showed that when you train animals on this tea maze, that initially their learning is under hippocampal control. Later on, it is under caudate control. So if animals are well trained and you remove the caudate, they're clueless. If they are not well trained and you remove the hippocampus, they're clueless. So here are different regions of the brain that are involved in the learning and expression of different aspects of the learning depending upon the degree of training. So Tolman was right. There is more than one kind of learning, and we can begin to look at the neurobiological underpinnings of that. So that period of time, about 1950, was really critical in our history. A lot of things happened. Donald Hebb published his book, The Organization of Behavior, in 1949. He was a student of Carl Lashley's. And in it, he proposed the dual trace hypothesis that you are familiar with. You can have an immediate trace which gets converted into a lasting trace because of the changes that take place underlying that. Remember the Hebb principles of the cells that fire together get connected. And that's what we usually think about when we think about Hebb. Hebb himself did not think that way, and he thought that was rather trivial because you had to have something like that, and that was sort of a throwaway early in his book. What he was really interested in is how systems in the brain get organized, and the principle that he had was to enable that to happen. You have to have a connection in there so that regions of the brain can begin to work together to generate behavior and solve problems. The other contribution that he made was that he was the teacher of Brenda Milner. Brenda Milner was his student, and he is the one who sent Brenda Milner to work with Scoville, and they are the ones who studied HM and a number of other patients as well, and gave us the critical findings from HM. A locus of the brain region that is critical for memory. New lasting memories are not formed if that region is gone. Remote and recent memories are spared, and skill at motor learning is not blocked. And that was in 1957. All of these things were found in a single patient, one patient. And I think if the author of Phrenology were around, he would have said, whoopee, this is what I said all along, that there are different regions of the brain that do different things and they cooperate in enabling behavior. And Brenda Milner gave us those findings, which have influenced the research of most of the people in this room. Now we go back to Gall and Spurzheim in 1815. I want to remind you what they said, having just listened to everything I've said, and I just ended up with Brenda Milner. It is certainly impossible to deny the mutual influence independent of the different brain organs. There is, however, a great difference between the correct assertion that the different organs exert a mutual influence upon each other and the incorrect one that each part does not exert its particular function in 1815. So we've gone from 1815 to 1957 through some tough times for the thinking and have come out with the same idea, but this time which is based on data. But Gall and Spurzheim had the ideas first. They didn't have the technology to do that. They didn't have the accidental discovery of a patient that would reveal things. They didn't have any of that, but they were right. Florenz was wrong. And Gall was correct. So 25 years after Brenda Milner's finding, we decided to establish the Center for the Neurobiology of Learning and Memory because we thought the time was ripe at that point for new discoveries. There are new technologies that you're all using today. There are new ideas that grew out of the findings that I've described. Everything was really ripe for the taking at the time. So we developed the center, and we attracted faculty, we attracted lots of graduate students and lots of postdocs, and we had a lot of fun building on the ideas that these people had proposed. And most of us here, maybe all of us here, are influenced by the ideas that I have discussed here. When you think back, many of you are working on specific brain regions. I've looked through the program. Many of you here are looking at the interactions of brain regions involved in the formation and execution of learned response. And I just hope that we will continue to think about the pioneers that influence the ideas that we are using today to generate our experimental findings. Folks, you are not novel. You're not novel. The ideas have been around a long time. You've just sort of sucked them in, and you're using them. And I think you should pay honor and respect to many of these pioneers who were despised in their time. And now I'm trying to revive them and tell you how important they were and how right they were, because they really are the pioneers that we respect. Now it's time to end this and say, using all of these ideas, it's time for all of you to go out there and go to work and tell us all about what you're finding, and I look forward to learning more from you, and thank you very much.